Good afternoon, everybody. Shall we begin for today's lecture?
Yes, sir.
Yes, sir.
Yeah.
Yes, sir.
Okay. So yesterday we had seen some mathematical equations pertaining to the techniques of centrifugation. I hope there were no problems in yesterday's lecture. Just a brief, you can say, recapitulation of yesterday's lecture. Okay, I'll just show you the slides. We have seen what is the sedimentation rate, the terminal velocity, and the sedimentation coefficient. Okay, so in today's lecture, we'll go for a brief description of the different types of centrifuge machines. Okay, am I clearly audible?
Yes, sir.
Yeah,
Yes,
today,
sir.
if you will look at the background, okay, my background is a little different today because I'm working from the comfort of my home. Okay, I'm not in the office today, right? And that is why I'm asking you whether I can be heard properly and whether you can see my screen. Okay, so the different types of centrifuges are based upon different factors. For example, based upon the size of the centrifuge, we can have small size centrifuge machines to very big models. Okay, we can have tabletop models which can be accommodated on a working table. And there are some models which are so large that they cannot be put on a table, but they are called as the floor models because we keep them on the floor. Okay, then based upon the volume of samples that can be used for centrifugation, we have different types of centrifuges. For example, the microfuge is the one where we have very small quantities of the sample material, and we normally use what are called as the microfuge tubes. Okay, have you heard about this word microfuge tube anytime? Let me just see whether I have any image of that. Yeah, this one. Can you see this one? No, this is not a microfuge actually. I don't know. I don't have an image here. Yeah, this one here. Can you see this? The first one?
Yes, sir.
Yeah, we also call it as an Eppendorf tube. Have you heard this name, Eppendorf tube?
Yes,
Yes,
sir.
sir.
Okay,
Yes,
so the instrument which makes use of these kinds of tubes, okay, they have a particular kind of a rotor, and that rotor can accommodate the Eppendorf tubes or the microfuge tubes, and therefore such instruments are called as the microfuges. Okay, microfuge. Okay, shall I think, shall I paste that figure? I just paste it here so that it will be good for your own reference. I'll crop it. Okay, can you see that now?
Yes, sir.
Okay, good. Okay, so these machines can accommodate a small amount of the samples, and the centrifuge, the field is normally about 10,000 G, or even sometimes it could be like 10,000 RPM. Okay, not more than 10,000 RPM, but it is less than 10,000 RPM. Okay, and then these are useful for separating bacteria. They can also be used for separating the blood components, especially blood cells. They can be used for protein purification or collection of the protein precipitate of the DNA, RNA, and so on. Okay, and then the only problem is that they do not have any cooling facility, and therefore if you need to employ cold conditions for the samples, then usually these microfuges are kept in a refrigerated room, okay, or in a cold room, and that will prevent high temperature from damaging your biological material. Okay. Then I have written a cost here, cost. So this I leave it to you. You can find out from your internet what could be the cost of these different instruments or centrifuges that we are going to study today. Okay. So that was the first type of a centrifuge, the microfuge. These little ones, okay, these are very small centrifuges. The cost of these is quite affordable. For example, they could cost anything from say 7 to 8,000 rupees to maybe about 15,000 rupees. Okay, depending upon how many vials can be employed, how many samples can be run in one run. Okay, they do not have any refrigeration facility, no cooling facility, and good for your small laboratory work, and they are portable also, so you can actually shift them from one place to the other. They're very lightweight actually. Okay, maybe the weight could be somewhat equivalent to your mobile devices. Okay, then okay, we have these large preparative type of bench centrifuges. Okay, although I have written here floor, I will delete this. That is not correct. I think I have to reduce the font. Yeah. Okay. So these centrifuges are slightly larger than the microfuges, and they can run from say 3000 to 700 G, that is the centrifugal field. And at the about the RPM, okay, I can say that they can have about say 15,000 RPM to 20,000 RPM speeds. Okay, so the figure here, the image shown here is actually the image of a rotor. Okay, and this particular rotor can be replaced. Okay, so we can change the different rotors depending upon the type of the sample or the amount of sample that we have. So for example, we can have say 5 to 10 ml of the sample in one bottle or in one vial, and up to say about 200 to 250 ml of the sample can be loaded in one particular bottle. Okay, so shown here in these images are the type of centrifuge tubes or bottles which are used. Even this one, I think you might have seen this kind of a centrifuge tube in your laboratory. Have you seen it anytime?
Yes, sir.
Okay. So,
Yes,
or
sir.
otherwise, when you come to the institute sometime after the lockdown, then I'll be able to show you the different kinds of centrifuge tubes which we use in the centrifuge machines. Okay, so depending upon the size of the tube which are employing, the rotors are different. Okay, we can remove one rotor from the instrument and we can put another rotor that we want. Okay, and this is a common type of the instrument which is used. It is a benchtop model or a tabletop model, and you can see some of the essential things. There is a lid here. Okay, the lid is lockable or it could not be lockable. For example, sometimes there are automatic lock systems. So when the centrifuge run starts, the instrument will lock up automatically, and then it cannot be opened while the run is going on. And only when the run stops, then the lid can be opened. But of course, if with that features, the cost of the instrument also goes on the higher side. But most of the instruments do not have this self-locking or auto-locking system. And these systems are present in the more sophisticated or the more advanced kind of machines. Okay. These are usually used for the separation of bacteria, separation of animal cells or other cell, even plant cells, or maybe protein, DNA, RNA, and so on, because the speed of the machine is say 15,000 to 20,000 RPM. Okay. And then these are the clinical centrifuges which I have shown here. These are very, very affordable, low cost instruments. They do not have any refrigeration facility. and they are normally used only for blood samples, okay, for the separation of RBCs or separation of WBCs, and even for the separation of serum or plasma, serum usually, okay. And the speed of these instruments is not very great, okay. They can run for something less than even 3000 RPM, but that is enough for your blood samples. Then again, they do not have any cooling facility, okay. So these are very basic instruments which are used in the clinical laboratory, right? Then one thing about this benchtop model again, this particular instrument which is shown here does not have a cooling facility also, but there may be some instruments which do have cooling facility. Okay, so I'll just write it down here. Cooling, okay, yes or no. Okay, so cooling may be available or it may not be available, depends upon the application, and it also depends upon the price factor. Is it okay? So then we go to the other type of instrument, and that is called as a high-speed refrigerated centrifuges. These are quite large in size, and therefore we cannot keep them on a table, okay, or we cannot put them on the bench. I don't know what was there. Okay, so these are kept on the floor usually. These are floor models. You can see the image here. Okay, and these instruments can be used for the separation of proteins. They can be used for the separation of cell organelles, and the centrifugal field in these machines is often to the level of about 1 lakh G. Okay, 1 lakh G. And in terms of RPM, they may go to about, say, 30,000 RPM, or maybe something less or more than that. Okay, so high speed and refrigerated centrifuges. This can also be used for differential centrifugation, a technique that we are going to see a little later, maybe in our next lecture. Okay, they can be used for separating nuclei, mitochondria, chloroplasts, and other organelles from the cell. Okay, they can also be used for the separation of microsomes, right? They can be used for harvesting bacterial cells, yeast cells, or even fungal cells from a large volume. Okay, and then they can be operated in another mode, which is called as the continuous flow mode. So what happens in continuous flow mode is that the medium enters the centrifuge device, the rotor, okay, or rather inside the separation chamber continuously. And as the cells are separated, the supernatant from the cells will automatically be collected continuously. There is an inlet and an outlet system which is provided, and therefore the instrument can be run for hours together. Okay, they may be run for about 4 hours to even say 8 hours to 10 hours. Okay, or sometimes even when I was working in a laboratory of Agharkar Research Institute. Have you heard this name anytime? Agharkar Research Institute, which is in Pune.
No, sir.
Okay, there is one institute called Agharkar Research Institute. It is a national level institute and funded by the Department of Science and Technology under the Ministry of Science and Technology, Government of India. So I was working there for my PhD, and even after that, I was working as a scientist in this institute. And there we had this particular kind of a centrifuge machine, which is called as a continuous centrifuge. Okay, and it was rather a normal floor model centrifuge, but it could be operated on a continuous mode also, because it had a special rotor which had an inlet and an outlet system. And we used to harvest the thiobacillus culture, okay, thiobacillus cultures using that instrument. And the problem with these cultures is that we get a very, very poor biomass in these cultures. Okay, so even if we centrifuge about say 10 liters or 20 liters of the medium, we normally get a few milligram only of the cells, okay, or rather few microgram of the cells. Okay, so that low quantity of cells are there, and therefore it is always preferable to go for a continuous kind of a centrifugation, And this centrifuge we used to run for overnight. Okay, we used to just switch on the instrument in the evening, say 5 o'clock or 6 o'clock, and then we used to go home or to our hostels. And then the next day at say 9:30 or 10 o'clock, we used to go back to the institute, and then we would harvest the cells, okay, collect the cells, right? So we can run it overnight. That is the interesting point about the continuous centrifugation. And then, well, these instruments also have some other additional features. For example, there is something called as the automatic locking system. So while your run is going on, the lid cannot be opened. It gets locked up automatically. And then if at all the lid is not locked, then the instrument display will show you a message saying that the lid is not closed properly, and therefore it cannot be operated. Okay. And then there is another facility which is also called as the imbalance prevention. So if at all your centrifuge tubes are not balanced, do you know what is this balancing of the tubes?
Yes, sir.
Yes, sir.
Yeah,
Yes,
what
sir.
is it? Why do we require that balancing?
So putting on the tubes on the opposite ends so that equal centrifugal force is distributed among the two.
Yeah, that is a correct answer. So normally what we do is we cannot use odd number of sample tubes. Okay, we always go for even number of centrifuge tubes, and these tubes are always kept diagonally opposite to each other. Okay, so the rotor has the places, there may be 6 slots or 8 slots or more than that. There is always an even number of slots. Okay, and whatever tubes you have, if for example I have only 2 tubes, then I would always keep them in a diagonally opposite fashion, so that the weight is balanced properly. And if the weight is not, of course I have to weigh each and every tube, and we should always take equal weights of the sample. Okay, the sample volume should always be equal, and that volume is in terms of the weight. Okay, and if for example there is an imbalance, the weights are not exactly equal, okay, then what will happen is there will be a load on the central shaft which actually rotates in the instrument. And if there is a load on the shaft, then there are chances that the shaft will break. Okay, and if the shaft breaks, then that is quite a very expensive thing to purchase. And lot of most of the time the instrument will be laid in the off mode for a long time, because it will really take a long time to procure a new shaft and repair the instrument. Is it okay? Well, so then we go to the next slide. That is the ultra centrifuge. Okay, so ultra centrifuge is a super high speed centrifuge, and here you see this number 6 lakh G. Okay, so the centrifugal force which is employed here is more than 1 lakh to about 6 lakh G. Okay, 6 lakh G. And in terms of RPM, I would say that the RPM speed of these instruments is about 1 lakh or so. Okay, 1 lakh or little more than 1 lakh RPM. Okay, so that is a very, very high speed indeed. And therefore, what happens is at these high speeds, if the rotor is going to move in a normal atmosphere, then there is a lot of chance that because of the friction with the environment, the rotor will get heated up. Okay, rotor will get heated up. And because of that, we need the cooling system. Okay, refrigeration system, for especially for biology work. This heating is not desirable because biomolecules or biological samples will get damaged because of the heat. Okay, so cooling system is always there. And another important thing is, in order to reduce the heating, these instruments also come with a vacuum system. Okay, so the rotor is placed in a chamber, and inside the chamber a high vacuum is normally employed, and therefore the frictional effects are minimized. Okay, friction with the atmospheric gases or dust particles, that is minimized. because of the vacuum. And in addition to that, we also have a refrigeration system. Okay. And then these chambers are also sealed and evacuated. Normally, the centrifuge tubes are also a special kind of a tube. So these tubes are actually sealed. Okay. And it is not just putting the lid on the centrifuge tubes and closing them. Okay. But because that also might lead to some sample spillage, that might lead to air going inside the centrifuge tubes. And therefore, these special vials are there which can be sealed completely so as to make them airtight. Okay. And therefore, when the samples are removed from the centrifuge machine after the centrifugation for collection of your precipitate or the supernatant, whatever it is, normally these seals are required to be broken. And therefore, these tubes are actually disposable tubes or disposable vials. Okay. So vials which are used for ultracentrifugation are always disposable type. You can use them only once because they are sealed. And for removing the sample, the seals are broken. The vials need to be actually broken. Okay. Then another important thing about these instruments is that the sides of the rotor, okay, at the rotor is somewhere here inside. Okay. And at the sides of the rotor, the walls of the instrument are very, very thick. That is called as heavy armor. Okay. Heavy armor means that the walls are quite thick. They are made up of heavy metal. And that is there for preventing the user, protecting the user. Okay. Because if something goes wrong, if there is some imbalance, or if the shaft gets broken in between, or somehow the rotor just comes off the shaft, okay, and it might break through the lead, okay, or it might break through the sides of the instrument, and some accident will take place, right? So in order to prevent that from happening, these centrifuge machines are also protected by means of heavy armor, right? Then they have auto temperature regulation. The temperature is always maintained to a certain level, plus or minus say 0.1 degree. So if you are setting the instrument at 8 degree or say 9 degree or 10 degree, then the change in the temperature will be only say 10.1 or 9.9. So 0.1 degree change in the temperature is allowed. But if the temperature difference is more than that, then that is not allowed actually. Okay. So that particular care is taken. And then there is a chamber locking system. Over speed control is also there. So your set speed and the actual speed of rotation will always be controlled. And there is imbalance prevention. So all these facilities are there in this kind of instrument. Then there are two types of ultracentrifuges. One is called as a preparative ultracentrifuge, where you can actually you can collect your sample. It may be proteins, it may be bacteria or viruses or whatever it is. And then these components which are separated, they can be further used for certain applications. So that is called preparative ultracentrifuge. This word preparative means exactly the same thing as we have seen in preparative electrophoresis. We have also seen preparative chromatographic techniques. Okay, so similarly here, so preparative ultracentrifugation means that we can collect the sample in some significant amount, and that can be used for your other applications. Okay, of course, as I told you, the vials are very small in size, so there is a limitation on how large a sample size can be used for ultracentrifugation. Okay, it is not a very large amount. It is a few, you can say a microliter to maximum about 1 ml or something like that. Okay, not more than that. Then there is another centrifuge machine which is called as the analytical centrifuge. Okay, and this is a centrifuge which does not allow you to have a large quantity of the sample for any other purpose. Okay, this is only used for analysis of the sample components. It is just like your analytical chromatography, or it is like electrophoresis. Okay, so whatever there are, the sample as it gets separated in the centrifugation, the different boundaries will be formed. Okay, and these boundaries are monitored by an optical system, like we have seen the Slyron optical system in what you have seen it. Do you remember?
Moving boundary.
In capillary, yeah, capillary electrophoresis, we have seen it. Okay, or moving
Yes.
boundary electrophoresis. So similarly, this particular optical system will help you to monitor the movement of the different boundaries of the sample as the sample components get separated. Okay, and that will also allow you to carry out some biochemical analysis of different biomolecules or biomolecule complexes. It may be, for example, antigens and antibodies reacting together, and when they react, they will form complexes, right? And if the complexes are formed, the movement of the complexes will be different compared to the free antigen or a free antibody. Okay, now that differences are also monitored by the optical system, and that will give you some information about, for example, in the last lecture we have seen about how we can use centrifugation technique to determine the molecular size of the molecules, isn't it? We have seen the mathematical expression, okay, or sedimentation coefficient. So that kind of information can be obtained from the analytical ultracentrifuges. Is it okay?
Yes.
Yes, sir.
Then we go to the next slide, where we have, this is a special type of a preparative ultracentrifuge, which may be run in a continuous mode or in a batch mode. Okay, so there are two modes in a centrifugation. There is a continuous mode and there is something called as a batch mode. Now, similarly, these two types of terms can be used also for your chromatographic experiments. Okay, I think I did not mention this when I was teaching about chromatography, but you can remember these two terms, batch mode and the continuous mode. So continuous mode is where you have continuous inlet of the sample. It may be in a chromatographic column or it may be in a centrifugation device. Okay, and there is continuous outflow or you can say outlet of the sample supernatant in the case of centrifugation, or it could be the column effluent in the case of a chromatographic column, right? Or it could be a batch mode operation where you put a certain fixed volume of your sample either in the centrifuge machine or in the chromatographic column. So for example, in the chromatographic column, you can put say 10 ml of the sample or 2 ml of the sample on the top. Okay, and then as the different sample components will get separated, you will then collect the fractions. Okay, small fractions you can collect from the outlet of the column. And then in the case of centrifugation, you can add like the normal centrifugation that you have already carried out in your BSc. Okay, that is the batch mode centrifugation, because a fixed volume of your sample is already put in the centrifuge tubes. You carry out centrifugation, and at the end of the centrifugation run, you just remove your tubes. You will find that the two components, that is the solid component and the liquid part, they are separated. Okay, the supernatant is either collected or it is discarded depending upon what is your application, and or that is the same with the case of the pellet. The pellet could either be discarded or it could be collected for your further use. Okay, now I think I have just 5 minutes to go, and if the meeting gets ended, then I think I would again have to restart it. So don't go anywhere. I'll continue for another 15 to 20 minutes after this particular meeting is over. Okay, in the second meeting. Is it okay for everybody?
Yes, sir.
Okay. So that was about the different types of centrifuge machines. How many types of centrifuges have we seen? I'll just go quickly through all the slides. Okay, we have seen this first slide where I told you that the types of centrifuge depend upon either the size of the centrifuge machine, or it could be based upon the maximum volume that can be centrifuged. Okay, there could be different types of centrifuges based upon the maximum speed of the rotors. For example, we have clinical centrifuges which run at something less than 3000 RPM. And then we have some centrifuges which can run up to say 20,000 to 20,000 RPM. And then there are the ultra centrifuges which can go to over 1 lakh RPM, okay, or about 1 lakh RPM. And then there are centrifuge machines which may have the vacuum system or they do not have the vacuum system. Especially this is the case for the high speed centrifuges, okay, or ultra centrifuges. Then there are centrifuges which can have a refrigeration system, that is to cool your sample, because at the high speeds, there is a possibility that a sample will get heated up. And then if it is a biological sample or if it is a heat labile sample, then that sample will get damaged because of high temperatures. And therefore, there should be a system to control the temperature within the set parameters. Okay. And then there are centrifuges which can be run on a continuous mode, because that is required because the sample that you have will contain a small amount of the solid material, insoluble material, which maybe you are interested in. And therefore, it will require a large number of batches to be carried out in the normal batch mode, and that is not feasible. And therefore, one goes for a continuous type of a centrifuge, where you just put your sample in a continuous fashion. Automatically, the sample will enter into the centrifugation chamber, okay, and automatically the supernatant will be collected, and therefore you don't have to really sit in front of the centrifuge and monitor the proceeding. Okay, so automatically everything will be done. And then there are preparative type of centrifuges and analytical centrifuges. Okay, so having seen all that, we now go to the next type of information, and that is about the rotors. I think the meeting is almost coming to the end. I don't know whether the time can be seen here somewhere.